all to today's academic session of the ICKD group. I think people are still joining, so we may just wait for a minute or two. I think we will start now. Good evening again, everybody. I am Dr. Vinod Kumar from PGI Chandigarh. For today's academic session, we have with us Dr. Vinod Sharma, who is Assistant Professor in the Department of Dermatology here at PGIMER Chandigarh. He did his PhD in nephrology from PGIMER Chandigarh and thereafter did his postdoc in the US, and after a brief period in the US, he has joined back as a basic scientist in the Department of Dermatology. So he has worked extensively on the topic of APOL1 and kidney disease in the United States, and today he will be sharing some snippets of this of the literature with respect to this. So over to you, Dr. Vinod. Vinod, looks like you are muted.
Okay, it's fine. Now you can hear me, sir.
Yeah, we do.
Okay. Good evening to all. So I'm Dr. Vinod, here going to present about the APOL1 protein and its role in the kidney function. So the role of APOL1 in our area or in our country or this continent is not that much relevant, because this protein is functional or having some association with the kidney disease only when it have some mutation in it that we call as G1 and G2, and that mostly belongs to the African ancestry. And as it has been reported previously that in India we don't have these mutation in the APOL1, but still it's very interesting protein, and we should know about this protein because it has evolved very recently and it has documented to have a very important role in the evolution of human beings. And till date, many mechanism has been proposed, but still very less has been known about this protein.
Vinod, your voice is breaking up. You may want to switch off your video.
Because of the
Can everyone hear Vinod?
Yes, sir, I can hear, sir.
No, we can't hear you properly, Vinod.
Okay,
Vinod, you might want to switch off your video.
Okay, sir. Okay, sir.
We lost you after the first.
Vinod, you may want to go back to the previous slide and maybe restart from there.
Sorry. Now
Yeah,
it's fine.
please restart. Yeah, that's fine.
Okay. So as we know that this APOL1 protein is associated with the kidney function, but its association is only because of the presence of mutation, that is we call as G1 and G2. But in our region, we don't identify these mutation in our population. But still, this protein is very interesting because it has been evolved very recently, around 30 million years ago, and it has been documented to have very important role in the evolution of human beings. And there are many functions which are defined about this protein, but still there are many undefined functions also which are revealing slowly with the new experiments. So around 10,000 years back, the Trypanosoma brucei, which causes the trypanosomasis in human beings, start evolving because the APOL1, which is present in the circulation of human beings, neutralize this trypanosoma and prevent its pathogen, its infection. So around 10,000 years back, it has evolved into two subtypes, that is a Trypanobrucei rhodesiense and Trypanobrucei gamiense. So what is that these two new species of the trypanosoma are able to infect the human beings and able to cause a sleeping sickness disease. So what happened that the Trypanosoma rhodesiense, it has developed one serum resistance antigen protein that react with the APOL1 and start its degradation and cause the infection to the human being. And in the Gambiaceae, they have proposed the one membrane protein, that is a TSG-GP mediated reduction in the membrane access. The presence of this protein in the trypanosoma will reduce the uptake of or entry of the APOL1 into the trypanosoma, and it can cause the infection. So after getting this trypanosoma infection, the APOL1 protein also start evolving itself so that it can protect the human beings from this infection. So around 5,000 year ago, means after the 5,000 year after the evolution of trypanosoma, there is a presence of or appearance of two mutation in the APOL1 protein that happen in the serum resistant antigen binding domain, because it is the domain which help in the neutralization of the trypanosoma, and trypanosoma bind to this the new species of trypanosoma bind to this and cause the infection. So there is occur a mutation in this region so that the APOL1 can do its function and can protect the human beings from the trypanosoma infection. So the individuals or the human beings who have the wild type APOL1, or they can get infected with the trypanosoma, but the human beings who have at least one variant of either G1 or G2, they are protected from the trypanosoma infection. But the human beings who have the two alleles, either G1, G2, G2, or G1, G2, they are also protected from the trypanosoma infection, but they are more predisposed to the kidney damage or kidney dysfunction. So if we, whatever the data is present available in the literature, that these APOL1 risk alleles, G1, G2 is associated with the preeclampsia, hypertension, FSGS associated CKD, albuminuria, and many other, including the HIV-induced FSGS. So most in the African region, it is a very high prevalence of the HIV-induced FSGS. And even in the African who have the ancestral gene from the Africa, they are also have the higher prevalence of HIV-induced FSGS. So if we look upon odd ratio is defined in this table. So the literature also says that sometimes that the presence of risk allele is not enough for to cause the kidney dysfunction or kidney disease. So there is a need of some certain secondary hits also, that might be either the presence of some other genetic factor like MY at 9 or some like HIV in some cases, or some environmental factors. So the whatever the if the individual who are having these APOL1 mutations are presented with the high protein loss in their urine, accumulation of interstitial fluid in the body, edema, and decline of the kidney function. They are also presented with lupus membranous nephropathy and collapsing nephropathy. In diabetic nephropathy, the role of APOL1 and its mutation is very limited because the diagnosis of these patients are not done based upon their biopsy. So they don't know that whether there is a change in the expression of APOL1 or not. But by the genotyping, it has been predicted that diabetic nephropathy is not associated with the presence of these risk alleles. So the patient or the individual who are having at least one genotype in the heterozygous form, they have the decreased renal function compared to the one who has both the alleles. So till now we have shown that, I have shown that the APOL1 risk alleles are associated with the dysfunctioning of protein, and they cause the kidney failure. But other people are now also hypothesizing that there is a positive side of these variants also, because Friedelman and Martin Pollack have said that nature selection determines which genes are successfully transmitted to the future generation. But the dramatic increase in the human lifespan over the past centuries have presented an unexpected challenge to our organs system, and presence or absence of a gene might be a step towards a longevity. And they have said that this APOL1, which has been evolving and helping in the humans to evolve, might have some role in the kidney function. So if we look at this APOL1 gene is located on the chromosome number 12, and it is the 6th gene of the APOL family, and its protein contains signal peptide, pore forming domain, membrane addressing domain, and the serum resistant antigen binding domain, where this mutation appears. So around 30 million year ago, because of the duplication, there were only 4 APOL members. That's the 2, 3, 4, 5, and 6. But around 30 million year ago, there is occurs the duplication in the APOL1, 2, and 3, and they have a result in the generation of this APOL1 gene. And if we see this chart over here, as the evolution started, and the human being are start fabricating from the other non-primates, the APOL1 is going to be disappear, and it retain only in the human beings. Like our closest relative, that is the chimpanzees, also don't have the APOL1, but only human beings have the APOL1. So if we talk about the role of role in the evolution, so people have calculated the number of mutation that has been occurred in all the APOL1 family. So they have calculated the ratio for a non-synonymous and synonymous mutation, and they have shown, because the APOL1 is not present in all except the human, so they couldn't calculate it for APOL1, but other APOL1, 2, APOL1, 3, 4, 5, and 6, they have calculated that if this ratio is more than 1, means they have a significant role in the evolution of the that particular species. So according to them, that the presence of APOL1 within that time in different human relatives help in their evolution and survival in the other environmental conditions. And the other thing is that if we talk about these variants, so if we look into the this APOL1 gene, so the presence of these 2 alleles make this gene more stable, so that it couldn't get some other recombination. People who have the G1, G2, their APOL1 gene or APOL family is more stable compared to the human beings or individual who having wild type APOL1. So this protection might help those individuals to evolve or go further during the drastic environmental selection processes. So if we talk about the G1, G2 distribution, so G1 variant is particular shows exceptionally strong differentiation in the frequency between population in the Africa. The rapid emergence of geographical differentiation again points towards some evolutionary forces, forcing an increase of risk variant frequency in some environmental conditions. Combined frequency of risk variant G1, G2 is much higher in the West Africa, where the pathogen they prudently protect against is at least currently limited to the East Africa. So what is happening that what the data of the Africa is showing that in the pathogen is more predominant in the East Africa region, what this genotype is more prevalent in the Western Africa. So they were saying that these two mutations like G1 to the G2 help in the killing of the pathogen, but the G1 mutation is protecting from human beings to get infected. You know, the G2 will kill a pathogen when the pathogen enters inside the human being, but the G1 is preventing that pathogen just to the trypanosoma to get enter into the human being. So the G1 frequency is increasing very drastically compared to G2 in the West Africa compared to the East Africa. So the scientists are predicting that it might be because of the evolution or the selection or the environmental selection occurring over on that region. So if we talk about the APOL1 in the circulation, because it will kill the trypanosoma when it will enter into the circulation. The liver is the predominant source of secretion of APOL1, and then second is the kidney. But the secretion of kidney, APOL1 by the kidney is not validated till date. So it is expected that it's only the liver produce APOL1 which is doing all these functions. And the APOL1 which is expressed inside the kidney is associated only with the kidney failure or kidney diseases. So there are many mechanisms which has been proposed in the literature that how these APOL1 risk variants cause the kidney disease and podocytopathies. They disturb the ions balance. They produce certain cytokines. They induce the lysosomal swelling, mitochondrial dysfunctioning. They do the autophagy blocks. They induce pyroptosis, and they disturb the cytoskeleton of the podocytes. And by doing all this, it will ultimately cause the podocyte apoptosis and death and the kidney disease. So regarding this function, there is also a controversy. Now the presence of APOL1 is controversial, but its function is also controversial. Some people, they believe that this function of APOL1 that is causing the kidney disease is a gain of function. The prediction depends upon the data which is they have generated through the in vitro studies. So, but the another group, it believes that it is the loss of the function, because if we look on the they says that if we look on the human biopsy sample, so there is no increase in the expression of APOL1, and if we look upon its inheritance, so it inherited in a recessive mode. So only those genes which are associated with the loss of function are inherited in the recessive mode. But in this, the people who believe that it's the loss of the function of APOL1, they have mostly in vivo data, but they don't have in vitro data to support this concept. But the people who are believing that is a gain of function, they have a number of in vitro papers and data that is showing that is a gain of the function. But in 2015, it was the first time when the role of APOL1 wild type protein has been defined, that it contains a autophagy controlling domain in its protein structure, and that will and that is the basic function of this protein. And after that, other people are start working on the APOL G0. So here I am going to discuss about the two role of APOL0, that is its role in the petal epithelial differentiation, and the other question is that does podocytes secrete APOL1 or not, because it is still not defined in the literature. So if we see the literature in 2015, Kersman et al. From the Germany, they have presented one paper where they have shown that the microRNA 193A regulates the transdifferentiation of human petal epithelial cells into the podocytes. And if we see the literature, it says that the petal epithelial cells don't have APOL1. So what we have tried to correlate these two molecules, that if the petals epithelials don't have APOL1, but when they get differentiated into podocytes, they have the APOL1 expression. So we check that we try to check that whether this APOL1 has any significant role during this differentiation process, or if it has, is it done through the microRNA or not. So first of all, what we did, we have checked the association of APOL1 and microRNA, and we find that the APOL1 is a very strong target of this microRNA, and if we overexpress this microRNA, there is a significant decrease in the expression of APOL1. So and if we overexpress the APOL1, there is a decrease in the microRNA expression. So we have defined a one axis of APOL1 and microRNA, that these both are complement each other. If we increase the APOL1, microRNA will go down, and if we increase the microRNA, the APOL1 goes down. So this all depends upon that which mRNA or which microRNA is present or highly present at that time. So to study this APOL1 expression during the petal epithelial differentiation, first we have checked whether petal epithelial cells really have APOL1 protein or not. So we asked those authors from Germany to send us petal epithelial cells, and we did the Western blot, and we compared with HEK. So these are the podocytes, these are the HepG2 cells, which are derived from the liver cells. These are the liver cells. So we got the APOL1 expression in these two cell type, but in these two petal epithelial cells and human embryonic kidney cells, we did not find any APOL1 expression. So then when we started to differentiate this protein into the podocytes, there is a appearance of the APOL1 expression, and it appears in different days of the differentiation. At the 14th day of the maturation, there is the highest expression of this protein, this APOL1 protein. And to check that this APOL1 protein is responsible for this PEC transition or not. So what we did, that we took the petal epithelial cells on the day 0 and overexpress the APOL1 G0 wild type only by using the plasmids, and we find that after 72 hours of overexpression of APOL1, there is a transition of PEC towards the podocyte phenotype. There is an increase in alpha-actinin, WT1, CD2AP, podoclexin, and there is a decrease in the petal epithelial molecules like PEX2 and claudin. So we can, by this experiment, we can say that the APOL1 alone has a driving force to convert the PEC into the podocyte phenotype. Whether they are mature or unmatured, that's very difficult to predict within 72 hours. So further to validate this thing, what we did, that we took the podocytes and then we silenced the APOL1 in these podocytes, and we checked the expression of PEC molecule, the petal epithelial molecules, and we found that with the knockdown of APOL1, these podocytes get de-differentiated and showing the phenotype of a petal epithelial cell. So by doing this, we can say that the APOL1 help in the petal epithelial cell differentiation, but at the same time it help in keeping the integrity of podocytes also. If we decrease this expression, they start de-differentiating into the petal epithelial phenotype. And if we transfect and if we go for APOL G1, G2 variants, so we will we have found that they are not able to convert this petal epithelial into the podocytes, and these properties are only intact with the G0, and G1, G2 are not able to do this transition. So by this work, we can say that the microRNA APOL1 are in very close proximity with each other. That if we decrease the microRNA, there is an increase in the APOL1, and there is an increase in the conversion of petal epithelial cells into the podocytes. On the other hand, that if the APOL1 goes down and there is an increase in the microRNA 193A, there is a more favorable condition for the petal epithelial cells. So as we know that these APOL1 variants are associated highly with HIV, and so we also tested the effect of HIV on this conversion process. So what we did that we took the petal epithelial cells and infected them with the HIV lentivirus, and we observed that these after 72 hours, these petal epithelial cells start showing the phenotype of the podocytes. Means the HIV can stimulate the differentiation of these epithelial cells to the podocytes, and there is an increase in the APOL1 expression. And when we silence the APOL1, the HIV is unable to do this differentiation there, and this shows that the HIV also works to the APOL1, and if we silence the APOL1, the HIV is not able to fully differentiate the petal epithelial cells into the podocytes, and this is a staining for different podocyte markers. So for this, to check it in vivo, we have took the control and the HIV in patient, and we have stained those kidney biopsies with the APOL1 and cytokeratin for the petal epithelial marker. So here we can say that the green is covering the petal epithelial cells, and this red one is in the APOL1. And when we co-localize these two proteins, there is a certain dots which are showing that in the HIV, and when there is a infection of petal epithelial cells with HIV, there is appearance of cytokeratin along with the APOL1. Means the petal epithelial cells are start showing the APOL1 expression along with HIV. So if we summarize this paper, then we can say that the petal epithelial cells have a potential to differentiate into podocytes, and this happen very efficiently in case of wild type APOL1 0, because it down regulated the microRNA and convert that petal epithelial cells into the podocytes. But in case of the risk variants, the petal epithelial cells couldn't convert completely into the podocytes, and they are in their transition phase, and that we call they are more in the more proliferative stage comparatively to the differentiation state. And if there is an infection of HIV, so there is a more accumulation of petal epithelial cells, because they are unable to convert into podocyte in case of APOL1 risk variants, while in case of the presence of wild type APOL1, these petal epithelial cells get transited towards a podocyte phenotype, and they are not going to accumulate in this area. So other function of the APOL1 is we have also shown that the APOL1 form a very unique complex with the adherens complex, because this complex is very important to maintain the podocyte integrity and the slit diaphragm. So by this work, we have shown that the APOL1 is able to preserve this adherens complex, and if there is a decrease in the expression of APOL1, this actin cytoskeleton get degraded. So for this, first we did some immunoprecipitation assays, and we have shown that APOL1 form a very close complex with the CD2AP protein. And we have done some fluorescence microscopy where we have shown that APOL1 co-localized with the CD2AP, and these are the reason on the periphery of the cells which are showing the co-localization with the APOL1 protein. And to check the destabilization, we have seen that in case of the risk variants, where is APOL1 G1, G2, there is a decrease in the CD2AP, nephrin, and dynamine protein, and there is no change in expression of dendrin. So this integration of the cytoskeleton is depend upon the translocation of the dendrin. It is hypothesized that dendrin, when it translocated to the nucleus, it will cause the transcription of cathepsin L, and that causes the degradation of the cytoskeleton. So in G1, G2, there is a more translocation of dendrin into the nucleus, and there is a more cytoskeleton disarrangement. And we also did some experiment with the microRNA, and we have shown that this stabilization of APOL1 mediated cytoskeleton is done through the microRNA. And we have done some animal experiments also, where we have stained the kidney tissues with the dendrin and nephrin, and we have shown that in the APOL1 risk variants, there is a more accumulation of dendrin in the nucleus, and that initiate the cathepsin L transcription and cytoskeleton degradation. So this was a summary of this work, that nephrin, CD2, AP, and dendrin, they are in a very close proximity with each other, and APOL1 is a part of this complex. So when there is a disturbance or there is a decrease in the expression of this protein, or this protein is not functioning properly, the dendrin get translocated into the nucleus, and it causes cathepsin L transcription and cause the cytoskeleton degradation. And then the other question is that does podocytes secrete APOL1 or not? So as we know that most of the circulating APOL1 is come from the APOL1, but there is no data about the secretion of APOL1 from the podocyte. It is believed that podocyte don't secrete APOL1 because podocyte predominantly express APOL1 V2 mRNA. So the APOL1 protein also have many mRNA splice variants. So there is a V2 splice variant which don't have the signal peptide. And for a protein to get secreted into the circulation, they must have a one signal peptide. So whatever the data is present till date, it is showing that the mRNA or the RNA splice variant which is expressing in the kidney don't have a signal peptide. That's why it remain retained inside the podocytes, don't come outside into the circulation. So there is a one paper in the 2021. So what they did, they have transplanted the kidney having the risk variant into a patient who don't have who have wild type APOL1, and in another patient, the patient is having risk variants where the transplanted kidney was wild type, and they analyzed the urine, and they found that the secreted APOL1 belongs to the liver, not from the podocytes, and they have confirmed that it comes from the liver, not from the podocytes. What our hypothesis is based upon the concept of the this domain, that is a membrane addressing domain. So we did a preliminary study where we tried to dock this protein on the plasma membrane, and we found that this APOL1 protein has a very significant binding property to the plasma membrane, and we hypothesized that the APOL1 is not secreted in the urine independently, but it comes along with the microvesicles or some other like exosomes, because it can integrate these molecules to their plasma membrane. So this work is still ongoing. So for this work, what we did, we have took the podocytes and overexpressed the APOL G0, G1, and G2, and then we have isolated the microvesicles by using the ultracentrifugation, and we have characterized those vesicles by using their protein markers and SAM or nanocyte analysis, and we evaluate them for the APOL1 expression. So what we observed that in case of G0, there is a we detected the APOL1 in all the microvesicle isolated from the G0, G1, G2, but its amount is more higher in the G0 compared to G1, G2. And if we see the podocyte and other markers, they are more loaded with the podocytes marker. So if we conclude this work, we can say that this APOL1 protein is not secreted independently into the urine, but it might comes with a microvesicle. So we have planned some more experiment of some proteomics analysis and all those things, so that we can get the confirmed result whether it is present in the exosomes or not. So at the last, if I conclude this all presentation, that the presence of APOL1 risk variants is not not that much relevant to us, because we don't have those risk variants. But the presence of this protein might be of worth, and we can study the function of this protein much better, because we are having wild type APOL1. And the existence of this APOL1 and microRNA axis is very new. And by targeting or entering with this axis, we can preserve the podocyte phenotype or podocyte upright left ciliate differentiation towards the podocytes. Other than this, the APOL1 is also documented to have some role in the immune regulation, and it also protects from the HIV infection by inducing the monocyte differentiation into the macrophages or decreasing the viral replication. Thank you.
Yeah. Thanks, Dr. Vinod, for a very, I would say, elaborative and yet crisp presentation on functions of APOL1, which in a way are not clearly known, but we do have literature which now, explores ways in which APOL1 could be related to kidney disease. So before I, throw the session open, Dr. Vinod, is APOL1, other than human trypanosomiasis and various forms of kidney disease, is APOL1, the high-risk variants, are they associated with any other form of human disease as well? Or these are the only diseases with which the high-risk variants of APOL1 have been associated?
Actually, sir, there is a data present available regarding the association with the hypertension and coronary heart diseases. That the patient who are having these G1, G2, they are more prone to develop these two diseases also. And there is also a controversy that whether hypertension associated kidney disease in the patient who are having these variants are because of like hypertension, or hypertension is because of the kidney disease. So it's still going on that which is coming first in these variants.
Okay, thanks, yeah. Dr. So with this, the session is open for comments, questions, and suggestions.
Good evening. Thank you very much for that presentation. I'm Dr. Gopal Krishnan.
Yeah, thank you, sir. Thanks, sir.
Fine. So it is really amazing to know that the nuances of the biological role of APOL1 induced protein, APOL1 protein, in various aspects of kidney function in health and disease. It is really fascinating. I think still a long way to go in this direction. But what is really perplexing is that the pathology which is classically associated with the nephropathic alleles of APOL1 gene, namely the collapsing glomerulopathy. In the published literature world over, but even in the Western world, in whomsoever they came across this collapsing glomerulopathy, they were able to identify the presence of the nephropathic alleles of APOL1 gene. But in India, we do come across collapsing glomerulopathy in different settings, including HIV and recently COVID and so on. But none of the Indian studies have shown that the presence of nephropathic alleles of APOL1 gene. It's really fascinating. Even though Professor Vivekant Jha has already published, studied on this subject and published that there is no existence of nephropathic alleles in Indians, we also wanted to do that in South Indian population. Just now we have concluded, we just looked for the presence of APOL1 gene nephropathic allele presence in 200 patients of ours, cutting across the entire spectrum of kidney disease, starting from collapsing glomerulopathy, FSGS, and CKDU, and SLE, and so on and so forth. None of them exhibited the presence of this. So we are just wondering whether it is only just APOL1 gene or a similar genes are also operating at the level of a podocyte in the parietal epithelial cell and so on. The stories, I think this fascinating story has just started. Thank you.
Yes, sir, it's very true, because this gene is also evolved very recently. So there are very high chances that there might be some other isoforms of this protein or this gene in our population. Because if we see our like MYH9, so this story starts with MYH9 in 2008 when they did the ADMIC study, where they studied the population of European African and American African, and they found that only the people who were having the African ancestry are having more prevalence of HIV associated FSGS, and they documented MYH9 as the prevalent gene. But later, when they analyze the data again, they find that MYH9, when they start developing the mice are not predicting, they are not telling the exact phenotype. So they have started to looking for some other gene, and they found that just next to the MYH9, this is the APOL1 gene, and it is basically associated with the African
exactly
ancestry. So might we have something else which is associated around, maybe on this chromosome 22 itself, nearby the APOL1 or somewhere else that need to be identified.
Exactly.
Yes.
Initially, we thought it was actually myosin heavy chain gene.
Yes.
Yeah.
Yes.
Thank you so much. Thank you.
Vinod, do you want to stop sharing the slides so that you can see everything?
Sure. So this is video, sir.
Yeah. No, you can switch on the video, of course. So, yes, thanks, Vinod. So one question I have is that you showed the association with MIR, one of the microRNAs. And so do you think there is any way for us to interfere with that microRNA APOL1 interaction, thereby preventing the development of disease?
Sir, sir, you want to say that if there is a disturbance in this axis,
can we do anything to this axis which will potentially affect the disease manifestation?
Yeah, sir, actually, in this work, we have tested the vitamin D itself. So, okay, what happens that the vitamin D regulates the microRNA expression. On the promoter of vitamin D, this is not published yet, but on the promoter of microRNA, there is a binding site for RXR. So, when the vitamin D binds to the VDR, it forms a heterodimer with the RXR and then bind to the microRNA and suppress its expression. And that's why the APOL1 goes up. So this is the one thing that we have studied, this one. But there might be some other molecules, like we can inhibit the microRNA itself if we want to increase the APOL1 expression also.
Yeah, okay. Thank you, Vinod.
And Dr. Vinod, another interesting, when we had looked up the literature from India regarding APOL1, then there is I think there is a family or there is a group of individuals who have been described as APOL1 null. And people have just looked at, whether those group of individuals or that family has kidney disease or not. Though We do understand that that is not the proper way to do it, but it's just reported in literature that none of the individuals who were APOL1 null had kidney disease. So this was just, and the authors then speculated then that it's likely that the APOL1 mutation, which is leading to disease, is not likely to be loss of function mutation.
Yes, sir. According to that was the paper who told that if the presence of APOL1 gene or protein doesn't matter, because if we don't have it, still we have a normal function. But the other people believe that it might be because of some isoform or some other supplemented genes. So that is helping in those individuals. Because if we talk about mice itself don't have APOL1 gene, but its kidney function is also good enough. So still, it's just in a controversial stage that what is the exact function of this protein and why the human being has this protein.
Any other questions from anyone? Dr. Varghese, Dr. Narayan sir?
Yeah, no questions. Very nice presentation and very intriguing to see all the work and the work that has gone in. And we look forward to more information. And like you said, we might know more about other standby or hidden things. Looking forward to that. Thank you.
Thank you, sir. Thanks a lot.
Right. Looks like there are no other questions. Vivek, then perhaps you can close the meeting.
Yeah. So thank you everybody for sparing time and joining this session. So we have had a very good session where Dr. Vinod explained about APOL1 and its association with kidney disease and provided some insights into how APOL1 could be related pathogenically to presence of kidney disease, specifically HIVAN and the collapsing glomerulopathy kind of podocyte disorders. So thank you, Dr. Vinod, for enlightening us on this topic, which is in fact which is relevant in the global nephrology community. So once again, I would like to thank everybody for. Thank you. Thanks a lot.
Thank you, sir. Thanks, sir.
Thank you so much.
Thank you.
Thank you, Vinod.
Thank you, sir. Thank you.